Yes, I'm currently doing my PhD in the laboratory of Christophe Luget and Vincent Formand. First of all, I want to thank all the organizers of this online series of hydrogenase seminars for giving me the chance to give a talk. I will present tonight a part of my PhD work that regards an electrochemical investigation of catalytic irreversibility into iron-iron hydrogenases. So first of all, I want to introduce the main character of this talk, which is the hydrogenase IDES from Thermoneurobac martrani, which is a very recently discovered hydrogenase by the group of Gustav Berggren in Uppsala. This hydrogenase strongly resembled the putative sensory hydrogenase IDES from Thermotoga marittima, and we will now see what are the similarities and the differences, and we will compare this group into the introduction with the standard IDA iron-iron hydrogenases that are very well known. So all these enzymes have the same kind of catalytic cycle that is probably very well known in the audience. So two iron atoms, CO and CN ligands, and this azo-dithiolate bridge bond to a cysteine to a 4 iron 4 sulfur cluster. Then at the N terminal of the amino acid sequence, we have an additional domain with iron sulfur cluster that delivers protons, which is also present into IDAS enzymes. But into this class of enzymes, we also have another cluster at the C terminal of the amino acid sequence here, that usually in IDAS from Thermotoga marittima binds the protein to an additional domain called PAS domain, which is thought to give what is the sensory function of these enzymes. Now, the PAS domain is actually lacking into IDES from Thermoanaerobacter martrani, which makes unknown right now the physiological function of this enzyme. Other important difference between IDS and IDA relates on the proton transfer pathway, which is a chain of very conserved residues into standard hydrogenases that drives proton towards and backwards the active site, which is completely lacking into IDS enzymes. This means that this class of enzymes have to have a completely different chain of amino acids to transfer protons towards the active site. That is still unknown. Moreover, there are two very conserved methionines here close to the active site of standard hydrogenases that are again replaced by other residues into the active site of IDAS enzymes. In the particular case of tamides, we have a serine and a leucine at the place of these methionines. Now, all these enzymes ideally catalyze hydrogen oxidation and proton reduction, and therefore they're suitable for protein film electrochemistry. In this technique, the enzyme is adsorbed onto the electrode surface. It is active and exchanges electrons with the electrode, and therefore we can measure a current that is directly proportional to the turnover frequency of the enzyme. And we can look at how this current changes as a function of different parameters. For this presentation, we will only look at how the current changes as a function of the electrode potential. We will also fit quantitative kinetic models to really understand what is going on into the enzyme when we change the electrode potential. Now, the typical experiment that is done to look at the activity of the enzyme as a function of potential is cyclic voltammogram. In this experiment, we measure the current as a function of electrode potential while we sweep it up and down within a certain window. The current, we measure a reductive current for proton reduction at low potential and hydrogen oxidation at high potential. When we measure at the potential at which we measure zero current, this is the equilibrium potential of the H plus H2 couple. Now, the current that we measure can depend on potential and time because it is the product of the steady state response of the enzyme that depends on potential and the active fraction of the enzyme that we have on the electrode that may depend on time. That is the case, for example, of the hydrogenase from Clostridium berginki that was presented by Alexei Silakov last session. This enzyme is known to have a strong inactivation at high potential, meaning that on the time scale of the experiment that we run, the active fraction of the enzyme decreases while we are going higher in the potential. Then we can reactivate this active fraction of the enzyme, this inactive fraction of the enzyme, by sweeping back the potential to a lower value. Now, when the active fraction of the enzyme change in the time scale of our experiment, it is detectable by a very different shape of the scan back compared to the scan forward, which leads to these strong hysteresis. These kind of catalytic shapes, we're not going to discuss them in detail in this talk. We will only focus on these kind of shapes here, in which the scan forward is exactly the same as the scan backward, meaning that the active fraction of the enzyme is constant over the entire duration of the experiment, and we only look at the steady state response of the enzyme as a function of potential. So in the title, I mentioned catalytic reversibility and irreversibility, so we need to introduce them in a couple of slides. Usually in enzymology, reversibility is linked to the concept of directionality. A reversible enzyme catalyzes the reaction in the two directions. But I would just call this property bidirectionality, and I would define reversibility in terms of thermodynamic driving force that an enzyme or a catalyst need to catalyze a reaction in either one or the other direction. To make this concept a little bit clearer, I do a mechanical example with a surface, a very smooth surface in which there is an ice cube so that there is no friction between the two. When the surface is flat, we are under equilibrium condition, then we can tilt it a little bit in either direction. And since there's no friction, the ice cube will fall down with a certain rate, meaning that a tiny departure from equilibrium leads to a movement of our object. This is what I would call a reversible system. On the other hand, if we have some friction between the object and the surface, like a pizza slice laying on a table, I can tilt the surface as before, and the pizza slice won't move because the friction force is still overcoming the gravity force that applies on the pizza. But if I tilt even further, eventually our pizza slice will fall down, meaning that we need to go much farther away from equilibrium condition before our object will move down with a certain rate. This is what I would call an irreversible system. Now, to study this into enzymes, we need to control the thermodynamic driving force we give to our system, which is exactly what we do in cyclic voltammogram, because when we change the electrode potential, we change the energy of these electrons that are into the reaction, and therefore we control the force, the driving force that drive the reaction in either one or the other direction. Now, standard hydrogenases, you can see that with standard hydrogenases, you need to go just a tiny bit away from the equilibrium potential to have a significant current, which is pretty much like the ice cube example I did before. And this is this makes this enzyme very reversible enzymes. On the other hand, in the case of tamidese, we need to go much farther away from the equilibrium potential before the enzyme catalyzes a significant current in other direction, which makes it pretty much like the pizza slice example. So this is a very irreversible enzyme. Of course, from the point of view of a catalyst, being irreversible is not desirable because this overpotential need is a waste of energy. And that's why it's interesting to study how nature can catalyze the same reaction in a reversible and an irreversible way to then understand what is behind and design molecular catalysts that can behave very reversibly. So tamidese is not actually the first irreversible hydrogenase we met, because the group of Fraser Armstrong in Oxford published in 2020 proton transfer mutants of two standard enzymes from Clostridium and Chlamydomonas that are very reversible. These mutants are two single mutants of these two glutamates. They were replaced to aspartate to increase the distance between the residues of the proton transfer chain. This increased distance leads to a decreased rate of proton transfer rate. And when you measure cyclic voltammogram, you can see here in black the wild type. It is very reversible. It cuts very sharply here the zero current line. And in red, for example, this mutant here, that cuts much less sharply the zero current line. This inflection here is much more pronounced, which is a sign of a decreased the reversibility or an increased irreversibility. It is the same. What was proposed to explain this difference is that in the wild type, the very reversible enzyme, the catalytic cycle goes through a proton-coupled electron transfer. So every time the active site gets an electron, a proton comes with it. And this leads to this kind of catalytic cycle that is called ordered, in which the same catalytic steps are taken clockwise or anti-clockwise depending on the direction of the reaction. To explain the increased irreversibility of the mutants of the proton transfer, it is proposed an uncoupling of this proton-coupled electron transfer, meaning that the decreased rate of proton transfer makes the proton not coming anymore together with the electrons. And this leads to a very strong conclusion, that is that the irreversibility of these mutants comes from actually two different catalytic pathways. One that operates at low potential in the reductive direction of the reaction, which is here in green, and a different catalytic pathway that operates at high potential in the oxidative direction of the reaction, here depicted in red. But what was shown from a work of our lab in 2019 is that this kind of catalytic cycle, the ordered one, with the same catalytic step taken clockwise or anti-clockwise, depending on the direction, can, of course, describe the reversible catalytic response, but it can also describe a very irreversible catalytic response. Just by having the value of these two parameters here, the catalytic potential, being very far away. So a large difference between the two catalytic potential means a large irreversible catalytic response. And if we bring them closer together to the point that they can also be inverted, then we're going to obtain a very reversible catalytic response. So tamides, being very irreversible, it is very tempting to use it to test if this kind of catalytic cycle can actually describe this irreversibility of tamides, or if we are maybe facing something much more complicated. And if this is the case, eventually, then we need also to understand where does the irreversibility come from. But before going to, to that part, we need to go a little bit back and remember that we're doing protein film electrochemistry. So there is one more electron transfer step that we need to take into account, called interfacial electron transfer step between the enzyme and the electrode. Indeed, what I want to show in this panel is that for the same values of the catalytic potential, these values, I can have a very reversible catalytic response in case of fast interfacial electron transfer. But then if I slow down the rate of interfacial electron transfer enough, this catalytic response will look like very irreversible, like the red curve. And this is not a property of the enzyme. This is just due to how the enzyme adsorbs on the electrode surface, which is something very hard to control. So we need to check if that is the case for IDES or not. To answer these questions, we can model the catalytic wave shape of tamides, which is what I'm showing here, and its first derivative at the same time, because there is more inflection into the experimental data that are very hard to see, and they are much more pronounced into the derivative. The model we use contains two redox steps, contains the assumption of an ordered mechanism, like this one, and fits also the rate of interfacial electron transfer. When we fit it to the experimental data, we obtain very good fits, meaning that this kind of catalytic mechanism can describe very well the irreversibility of tamides. We don't need to invoke a much more complicated scheme. And a parameter that we can obtain is the difference between these two catalytic potential, which is actually 300 millivolt, pretty large. We now need to check if it is the real reason of the irreversibility or if we're facing some interfacial electron transfer rate, slow interfacial electron transfer rate. And to do so, we can play with this value and we can force it to be smaller and smaller. So as I said before, when the difference between the two catalytic potential is small, the enzyme is basically intrinsically reversible. And then we can fit our data with this constraint and see if we can compensate for this constraint with a slower rate of interfacial electron transfer. When I fit with this smaller delta I cut, I cannot obtain very good fits, meaning that this large difference between the two catalytic potential is actually really needed to describe this irreversible shape of tamides. And therefore, tamides is really intrinsically irreversible. So to sum up this first part, we showed that an ordered mechanism can account for a very reversible shape and also for a very irreversible shape into hydrogenases. And the irreversibility of tamides is actually coming from this large difference between the two catalytic potential. Now, this first part we could have published before, and what I want to show in the second part of this talk is some ongoing work that we are doing to try to understand actually where does the irreversibility comes from, or in other words, why IDES has this large difference between these two catalytic potentials. And to do so, we need to go a little bit deeper into the meaning of the catalytic potentials. They are not the redox potential of the active site. They do depend on it, but they also depend on the rate constants that are into the catalytic cycle, into the chemical steps of the catalytic cycle. And this means that into the value of the catalytic potential, we have information about what is going on in the catalytic cycle. And we can use it by measuring the catalytic potential as a function of another parameter. And for example, we did it as a function of pH, so proton concentration. What I show here is the catalytic potential as a function of proton concentration for tamidase, the reversible enzyme, in circles, and the very reversible enzyme from Chlamydomonas renarditi in squares. And in the bottom panel, I just show another parameter, which is the limiting current that I can obtain from my fitting procedures. That again depends on the rate constants of the chemical step. So it is just another parameter I can use to understand what is going on into the catalytic cycle of hydrogenases. So to really gain information from this plot, we need to have a model that describes how the catalytic potential and the limiting currents depends on proton concentration. Therefore, to build this model up, we can work on these equations by assuming that at least 2 of these 4 steps have to depend on proton concentration. Okay, yeah, these are the first step, the 4 steps, so the 2 redox potential and the 2 chemical steps. But we're not starting from scratch. We do not have to try all the combination, because actually, thanks to a work done in the laboratory of Wolfgang Lubitz in 2017, they showed that for Chlamydomonas renarditi, one protonation step is coupled to the first redox step at low pH and to the second redox step at higher pHs. Low and high relative to the pK of this one electron reduced state of the active site. So we already know where is one protonation happening. We need to find out where is happening the second one. It can either be coupled to the first chemical step, and this gives model 1, or to the second chemical step, and this gives model 2. Now, independently on where we assume this second protonation to be, we need to remember when we model it that we have a proton transfer chain of residues that actually can work as a proton relay. This proton relay can have a pK, and so the way we measure this rate constant for protonation and deprotonation is to assume that the protonation rate constant here is maximal at pHs lower than this pK of the proton relay, and then it decreases 1 decade per pH unit at pHs that are above this pK, and vice versa, exactly the opposite for the deprotonation rate constant. So if we modify the equation I showed before of the catalytic potential with these assumptions for the protonation steps, we obtain equations that looks like this, that are exactly the same for model 1 and model 2. So it is exactly the same equations independently on where we assume the protonation to be. What changes between these 2 models is the meaning of these apparent acidic dissociation constants that, again, they depend on the pK of the relay of the protons and on the rates of the chemical steps into the catalytic cycle, in a different way if we assume a protonation to be coupled to the second chemical step or to the first chemical step. Now, these parameters of the model are complicated and very hard to compare between different enzymes, for example. So the parameter we're going to really look at in the next few slides is the pK of the 1 electron reduced state of the active site, because it's a parameter that also appears into the equations, and we can really use it to compare different enzymes. So when we fit our catalytic potential and limiting current with these models, of course, both the model will fit equally well the data because the equations are exactly the same. What changes is the value of that pK I was mentioning before. We can see that if we assume a protonation coupled to the first chemical step, we have a neutral pK, while if the protonation is coupled to the second chemical step, the pK is much more acidic. And now we can compare this information with the same parameter measured by other techniques. For example, spectroelectrochemical titration. Again, from the work I mentioned before, it was shown that the pK for Chlamydomonas renarditi, it is neutral, which is fully consistent with a protonation coupled to the first chemical step. On the other hand, again, in Mulheim, in the laboratory of Wolfgang Lubitz, it was shown 1 year later that sensory hydrogenases have a lower pK than standard hydrogenases, which is fully consistent with a protonation coupled to the second chemical step rather than the first. So now I will just try to sum up everything In the first part, we have shown that reversible and irreversible hydrogenases have the same kind of catalytic cycle, ordered, in which the same chemical catalytic steps are taken clockwise or anti-clockwise, depending on the direction of the reaction. Then, to understand the difference between reversible and irreversible hydrogenases, we went further into the analysis of electrochemical data, and we compared them with spectroscopical titration. And for the reversible enzyme of Chlamydomonas and the irreversible one of Tamides. And we could actually conclude that it is the sequence of the step to be different into this catalytic cycle between reversible and irreversible enzyme. So in particular for Chlamydomonas renarditi hydrogenase, we obtain something very consistent with spectroscopy, in which the catalytic cycle can be composed of one protonation step coupled to the first or the second redox step, depending on the pH, and another protonation step coupled to the first chemical step, which makes very likely to have hydrogen binding and release coupled to the second chemical step here. On the other hand, for IDES from Thermonarbacter martrani, it is less clear for the moment, but we have a protonation that is coupled to the second redox step, and a protonation, the second protonation that is going to be coupled to the second chemical step here. This leads to two hypotheses. The first one assumes a hydrogen binding and release step in the first chemical step of the catalytic cycle, and this makes HOX to be the catalytic intermediate between the two chemical steps. And therefore, we would have an already protonated state of the enzyme that is going to be reduced and accept the first electron. But since these states were never detected by spectroscopy for IDES enzymes, this hypothesis is probably less likely to be the right one. On the other hand, the other hypothesis assumes that we can imagine that, for example, when the second proton comes into the catalytic cycle, that suddenly hydrogen is formed and released. This would make HOX to be the first electron acceptor. It is then reduced to this species, and then can be that are unprotonated, and then can be further reduced and protonated to form two species that have two electrons and one proton that are linked by a low chemical step. Now, since these two species were never detected by spectroscopy, it's very hard to predict what is going on into this part of the catalytic cycle. Therefore, we surely need some more investigation through spectroscopy and electrochemistry to really find out what is the catalytic cycle of irreversible enzymes. But for the moment, at least we can propose that reversible and irreversible hydrogenases have the same kind of catalytic cycle, in which is just the sequence of the catalytic steps that change between reversible and irreversible enzyme. So with this, I'm finishing. I would like to thank my team here in Marseille, particularly Christophe and Vincent, that helped me a lot with this project, and they taught me a lot during these years. Gustav and Heinrich from the Uppsala University for the collaboration about the Tamides project, and also James and Olas from Max Planck, even though I know James is not anymore in Mulheim, for all the discussion and the help with the spectro-electrochemical analysis. And all of you for your attention, and I'd be happy to take all the questions.
Thank you, Andrea.
Thanks.
Great talk.